Good afternoon, everyone. As Flaminia has kindly explained, there are three of us presenting today. I'm going to take the lead, but Steph and Dave will help with questions as we go along and at the end. So I'm going to talk about the UniProt knowledge base, but specifically focusing on proteins which are involved in antimicrobial resistance, or AMR, in some way. What first is UniProt? It's a comprehensive, expert curated, publicly available database, and it includes protein sequence, function, and variation information. And I'm going to give you a brief introduction to the UniProt website and then highlight some of the resources which should be of interest to people who are working on AMR at various stages in their career. So I'm going to start by talking you through the content of the presentation. I'm going to say a tiny bit about AMR. I'm sure many of you know a lot about this already. Then introduce the website, talk about the functional data that's in a particular protein entry, using one as an example, how to look for proteins which have a role in AMR in the UniProt website, look at the function data and the structural data for that entry, explain a little bit about some of the analysis tools that are on the website, how you can get at the protein sequences themselves, and then finally take you through some examples of specific proteins which have been curated by UniProt, which play a role in AMR, including some which are indirectly are involved in AMR. So, for example, in biofilm formation, and then round off by saying a little bit about how we add more value to the data, which may be useful to people working in AMR. Excuse me. I don't really need to show you this, but as a reminder, I thought I should say a little about how important the threat is posed by AMR. It's believed to have killed over 1 million people in 2021. Most of these numbers come from a Lancet review, a systematic analysis, which was published I think in 2022, and covered a very large systematic review of data worldwide. AMR is associated with nearly 5 million deaths, or it was back in 2021. And again, as you probably know, its AMR rates are higher in many low and middle income countries. This summarizes some of the organisms which are prone to developing resistance as specified by the World Health Organization. This was published only a couple of years ago. And as you can see, there's a long list of high priority species and groups of species. So getting back to an introduction to UniProt itself, as I've already said, it includes sequence and function information. It's comprehensive, it's of a very high quality, freely available, and we aim to release an update to the data every 8 weeks. Some of these deadlines are not cast in stone. It includes tools for analysis of the protein sequences, and one of the almost unnoticed, to some extent, great powers of the UniProt website are the links to other resources. There are a massive number of links to other databases. I've mentioned function. It also includes subcellular localization information, information about 3D structures and protein interactions. And this data is produced on the website via several different pipelines. There's an automatic annotation pipeline which creates entries which have been annotated colloquially known as tremble entries. Then there's a subset which have been manually curated by expert curators here in the team at the EBI and at our collaborators in Geneva and in the USA. Then on top of that, there's also a machine learning aspect to some of the annotations created in UniProt. At the moment, these only include names which have been predicted by the Google Prot NLM pipeline, but this is going to be augmented soon. All of our data is available programmatically, and we also encourage annotation or curation by the community, and we have various entries which have been annotated by scientists from the wider scientific community outside UniProt. You may be wondering where all the protein sequences come from. Well, the vast majority come as submissions via the nucleotide sequence databases. So ENA is the database that holds these at the EBI, which then is part of the wider international nucleotide sequence database consortium. But there are other sources. Ensemble, the genome database and browser here at the EBI, the protein database, PDB, another worldwide consortium, which of course will include structural data as well, and a few sequences which are submitted directly as protein sequence themselves from the wider scientific community. And these are all imported and go into a single database, which initially is made non-redundant by a process of merging. And I'll say a little bit more about that next. So I've said where the data comes from. The initial database called UniPark, which consists of a non-redundant set of sequences. These at this point are not annotated. And then they then enter a couple of different automatic annotation pipelines. Shown here is UniRule and Arbor. They go through a process whereby they are then taken up by expert curators, a subset of those sequences, and then based on data from the literature, they are reviewed and then updated and returned into the wider UniProt database or knowledge base. Let's take a look at some examples. I'm going to talk about one particular example, and because we're interested in AMR, I'm going to use this protein, a beta-lactamase. So this degrades the very wide set of antibiotics which belong to the beta-lactam family. So many of you will have heard of these, I'm sure. It's a class A beta-lactamase. There's 4 classes. So I'm going to talk a little bit about CTXM1 and use this as a way of guiding you through a protein entry in UniProt. So here I show you the top of a web page which is for this particular protein, this entry. And down the left-hand side here in this purple box, you can see that I've called this the sidebar. So what this allows you to do is to navigate through the whole entry without having to do too much scrolling. So there's all kinds of information down the left-hand side here in this sidebar. But then another way of viewing the content of an entry is in this series of tabs here, again outlined in this purple box. On the left here, you can see it's highlighted or underscored entry. So that's the main view of a protein in UniProt in the website. And then across to the right here, we've got something called the feature viewer. So this allows us to see view the data in other ways. And then highlighted in red here, this is a description of the function of the protein. And an example of this kind of functional information is shown in this next slide. Because this is a beta-lactamase, it's able to degrade the general class of beta-lactams and then produce water and a substituted amino acid, a beta amino acid from that antibiotic. And this is a set of data which may have been added automatically. In this case, I think it was, because it derives this little tag here, shows where the data came from. So the fact that it's got a gold little bookmark here, this is meant to indicate that a human annotator has checked that this makes sense. This is appropriate for this particular protein. And this information, this view also includes things like an EC number from the Enzyme Commission, and tells you a publication, if there is one, associated with the data. And then it also takes you to links to external databases like Rhea and KEGG. Rhea is a chemical database which maintains the detail of chemistry. So in fact, this is pulled in from the Rhea website. And KEGG maintains information about the chemicals themselves rather than the reactions. This is scrolling further down the entry. This is an example of the kind of information you can see associated with the protein function. It's got a molecular function of a hydrolase, which fits with it being a beta-lactamase, and it's involved in antibiotic resistance as a biological process keyword. And then there's various other bits of information such as synonyms or alternative names. So I've called it beta-lactamase CTXM1, that's the recommended name, but it's also referred to as, in the literature, as kefataxamase or beta-lactamase MEN1. There's some publications which have called this particular protein by that name. Scrolling further down an imaginary web page for this entry, this shows you that this protein has several structures available, including a predicted structure from AlphaFold. And then if we move away from the entry view that I've shown you before, this is the feature viewer I'm going to show you next. And here, this is the active site of this particular enzyme, and gives you a little bit of information about what kind of catalytic activity it's able to perform, and the fact that this is a key residue in the active site at position 73, and tells you where the evidence came from. It's by similarity. It's actually by being similar to another member of the beta-lactamase family. And this gives you a link to that other particular enzyme that's used as the model or the template for that activity. The other way of viewing, scrolling down through the feature viewer, you can see there's other information as well as active sites. There are things like the degree of coverage in a structure and other structural features. So for example, these are different types of secondary structure in that particular protein sequence. Another important type of information that's found in entries is where there's known membership of a protein in a wider protein family. And this has been manually curated by one of our expert curators. Whereas some of these other family and domain database memberships have been added automatically. The sequence, as I've mentioned at the beginning, near the beginning, can be downloaded, and there are various other keywords associated with an entry, and then links to external databases which hold nucleotide and protein sequences. And towards the bottom of an entry, there's always information where available about other similar proteins clustered in various ways. So identical proteins at 100% identity, and then other thresholds too. And I've mentioned sequence. Well, one of the things you might well want to be able to do is to look to find similar proteins. And there's a BLAST tool built into the website, which allows you to take either an entry or your own sequence of interest and carry out a BLAST search. You can do multiple or single sequence BLAST searches. which need to be in FASTA format. And you can then restrict those results if you wish to by taxonomy, using this box here. As an example, I've done a search with this protein, which is the one we were talking about at the beginning. This is its ID. Down the left-hand side here of this result, you can see there are different filters which allow you to decide which particular subset of the hits you're interested in. So, for example, if you're only interested in other entries that have been reviewed, or which other organisms they might come from, or related, or yes, which other similar sequences are found in other organisms. And another view of the results is if we take this particular entry here, that's this hit rather, which is inside this green square. Below here, you can see the alignment of those two sequences, the query and the result of the search. And then let's say you find some interesting looking hits and you want to look in more detail at the relationship between those sequences. It's possible to select by clicking on tick boxes down the side of the list of results, and then feed those into a multiple sequence alignment. And this is an example of this kind of thing. You can put multiple sequences in from your own choice or from the BLAST search. And then this is a display of the kind of thing that you could see from a multiple sequence alignment. And one of the things that some people find very useful, I do it certainly, is highlighted here in green. You can see the annotations themselves highlighted or brought to your attention as shown here for these sequences in the multiple sequence alignment. So in this particular case, I've chosen to look for the active site of the related proteins. And you can see this particular residue, which is at the active site, is very highly conserved. It's identical in all of them, which is unsurprising because they're all beta-lactamases. Moving on to other kinds of proteins, other classes of proteins involved in AMR. I'm going to talk about a few examples which are either known or predicted to play a role in AMR, and they've been curated over the last few years, either by myself or by others. It's not meant to be, this slide's not meant to be a comprehensive list of AMR mechanisms, just some examples of the classes of proteins which have been curated and either involved in AMR or in some cases could be models for potential antimicrobials. So for example, down the bottom here, there are a couple of classes of, or a few classes of host protein, which, such as the defensins, which some people believe may be models for future drugs as antibacterials. And likewise, bacteriophage have been used in themselves as an antimicrobial system, but I'm not going to say any more about those. First, I'm going to talk a little bit more about beta-lactamases, and then an example, well, beta-lactamases in general, which I've annotated quite a few, and also say a little bit about an example of an efflux pump. So this is a system which actively removes a wide range of molecules from a microbe by transport out of the cell. Why did we choose beta-lactamases? Well, they're a huge number of proteins which belong to this class of enzyme. They are very well annotated using the automatic pipelines in UniProt, but at the time that I first got involved in this, some of the annotation, the manual annotation, was a bit patchy, and so I made a decision to try to look for those proteins which were not very well annotated and or were perceived as having a high clinical importance, mainly based on reading some relevant reviews and also getting advice from colleagues. Going back to our example that we used earlier on, CTXM1, it was one of the proteins that was not particularly, I think it was manually curated, but it wasn't particularly annotated in detail. And one of the things that we did was we were able to show that, or rather, no, to use data from the published literature, which showed that it could hydrolyze both penicillins and cephalosporins, including a particularly important drug called cefotaxime, which was often used. This information is probably a little bit out of date now, but was used as one of the more last resort choices in therapies. And less important, but from the point of view of a curator, very important, I tried to rationalize the way the numbering of amino acid residues was handled by the literature, which I found a bit confusing. So moving beyond CTXM1, these were some of the enzyme groups or families which are in the clinical literature, which, according to various reviews, including this one by Bush and Jacobi, were seen as having high clinical importance. And I've annotated several members of these groups. And although I've only listed one example for them here, there are usually several which were chosen from the group as representative. And this column here called molecular class, this is one of the ways in which the beta-lactamases are clustered or grouped together by sequence similarity, essentially. So there are 4 groups, A, B, well, there are also B2 and B3, I believe, A, B, and C, and D. And the most diverse group are the group D, and they were at one of the last groups that I did work on. I mentioned efflux pumps. They're particularly important in AMR because they can actually export a very wide range of xenobiotics, including antibiotics. So they give an advantage to the organism. This is an example of one that early on I was curating. It consists of 3 proteins in a complex which sits in it crosses both the outer membrane and inner membrane of bacterium, and it has quite an interesting structure. It's found in this particular case, this is from Pseudomonas aeruginosa, but there are very similar complexes. It's possible to see the probably evolutionary shared origin of them in other species. So in E. coli, there's a very well-known pump called ACRAB Tol-C, which is highly similar to this structure and to these proteins in Pseudomonas. And what we were able to do was to improve the description of the structure and its likely mechanism, and include information about the very broad substrate specificity. And one thing that was very interesting was that there are a class of drugs called pyrimidine derivatives, which are quite effective inhibitors of the efflux pump. I'm not sure what their status is in clinical terms at the moment, though. Moving on to another class of protein which have an indirect role in AMR. And some people have described these kinds of proteins as belonging to a secondary resistome. And what they've proposed is that they might be a source of antimicrobials which could act in almost as adjuvants or as helpers to use together with the classical antimicrobial drugs, for example. And I'm going to say a little bit about one protein, actually two proteins, one which is involved in biofilm formation and another one which is more unexpected. So this is a protein called BRLR. It's a regulator of transcription, and it again is from Pseudomonas, but it is highly conserved. It's found in other species. And at the time I started work on it, it hadn't been annotated at all, except by the automatic annotation pipelines. And so I added papers to it, including some structure papers, and it seemed clear that this protein was responsive to a known bacterial second messenger chemical called cyclic di-GMP, which you can see here in this diagram, which I've lifted from a review, which unfortunately I haven't included a reference here. I should do that. It's circled in red here, BRLR, and one of the reasons it's important for AMR is, as well as being involved in the formation of biofilm, it regulates several classes of gene which are involved in either removing or, in fact, they are involved in removing xenobiotics like antibiotics. So the efflux pumps I've just been talking about, and also ABC transporters. Some of these are also involved in exporting xenobiotics. So both of these probably are involved in making cells more resistant to antimicrobial agents as a result of the activity of this transcription factor. So it activates those classes, but it also has other roles as a repressor. So it represses another completely a different system of proteins called Fo-PQ, which are involved in, amongst other things, phosphate metabolism. And so overall, this transcription factor plays a role in AMR as exhibited by biofilms. Excuse me. Something more unusual, which is a bit of a stretch, really. This is another enzyme, but it's a dual function enzyme. It's both an aldehyde and alcohol dehydrogenase. It's conserved in various organisms. And it's not directly involved in AMR, but it does, if it's knocked out in bacteria, it enhances the susceptibility of, it enhances virulence in a mouse model, a mouse infection model. And perhaps even more interesting, this FDA-approved drug called fomepizole, shown here on the right, it's approved. It's an inhibitor of alcohol dehydrogenase. I believe it might be used in treating alcohol abuse or alcohol susceptibility problems. And obviously this is not an approved use, but apparently at least in the animal model, it enhances susceptibility to antibiotics. So if you were to use it together with an antibiotic, it could potentially act like an adjuvant. Those are my main examples of proteins which have been curated. It's only scraping the surface of the wider set of proteins that UniProt have curated. Now I'm going to say a little bit about other ways in which UniProt adds value to the AMR researcher community. So I mentioned right back at the beginning that we have very many links to other databases, and that's important. But the other thing that I didn't mention is that part of the process of manual curation is that we often will identify clear orthologs of proteins that we've annotated, And the annotations from those entries are then propagated. That's our own jargon word, meaning we transfer the annotations to those orthologs. And the kind of information we'd propagate would be protein names, functions, catalytic activities, and so on. And so the other thing is that as well as the manual process of propagation, these manual entries then act as templates for automatic annotation. So they're entered into the automatic annotation pipelines. And I just want to draw your attention to one particular knowledge base or other database, which our data is, we have links to these databases for our entries. And that is CARD. And I'm going to say a little bit more about CARD. So CARD is the Comprehensive Antimicrobial Resistance Database. It's widely used, and we have now got links between UniProt entries, the reviewed entries, and also an unreviewed set, which is much larger, which have links to a CARD entry. So in this example, in the screenshot on the right here, you can see this is on the left, I'm just circling, this is a UniProt accession number and the species, the name of the protein. And then on the right here, you can see there's a cross-reference or a link, a hyperlink to the relevant CARD entry. So, and it tells you a little bit more, as well as if you were to click on this ARO colon ID, that would take you to, as you can see, this little icon indicates, it would take you to the CARD entry. And we also carry a little bit of information about the resistance mechanism. So in this case, this antimicrobial, sorry, this enzyme works by inactivating antibiotics. And we're hoping that, or at least this is what we're expecting, sometime this summer, we're going to get reciprocal links back from CARD to our entries. So as well as being able to get to CARD entries from our database, you can then do a search or browse through the CARD database and then find a link back to UniProt. I mentioned keywords earlier on. I'm not really going to say any more about this other than to say that one of the ways in which we add value is, and this is built into the automated annotation pipelines, keywords that we add manually to entries are then propagated to other entries, to unreviewed entries. And this gives you an idea of the scale across this bar chart here. There are far more unreviewed, if we go in this light green bar here, far more unreviewed entries from bacteria, which carry a keyword for antibiotic resistance or virulence. The green one is antibiotic resistance and the yellow is virulence. So to summarize, I've given you a whistle-stop tour introduction to UniProt and said a little bit about some of the annotations that have been created manually and automatically in entries in UniProt, and how some of these can be helpful for AMR research. We would love to hear suggestions from you for any other potential target proteins for future curation. Give you an idea of the scale of the effort involved in UniProt. It's a very large consortium spread across 2 continents, at the EBI, the PIR in the States, and SIB in Switzerland. And our main funding comes from the NIH, but we also receive funding from ENSemble, Open Targets, and from the Swiss government. And if you want to find out about future ENSemble EBI webinars, you can go to these links, which you'll find also in the presentation when you receive it. And at that point, I'd like to stop and then take any further questions. Thank you.
Thank you so much, Paul, for a very interesting presentation. I think what I really liked about the way you structured this presentation was that you not only provided all the information, but also was a little bit of a journey in your job as a curator and how you created these proteins. And it was a little bit of a story. And I think it also reflects on the questions that we got from the audience, because we had many, many questions about actually the curation process. So I can see that David and Stephanie already replied to some of that. So maybe we can start from the ones that are still open. And then if we have time, we can also go through the ones that had already received like a written answer so we can share more widely. The first question that has received quite a few votes is, how do you decide how to prioritize what protein group you want to create first out of the many that you could do?
That's a mixture of different things. Some of it is reading reviews, literature reviews, and I also, when I first became involved, I had conversations with other experts at EBI, at the Sanger, and based on people I'd met in my past career. So for a while, I worked on a project looking at susceptibility to one particular organism, Streptococcus pneumoniae, in a mouse model. And so I'd made contact through that with other researchers. So I asked some of them. So it's a mixture of different things. I don't know if David or Steph might have anything to say about that as well.
I answered a similar question from one of the other attendees, and I emphasized that we've got a broad range of expertise at UniProt, and each curator has their own field of interest. So they have different ways of selecting what they prioritize. But one of the things that I like to emphasize is we really appreciate when the community sends us a request. So we don't always have an overview, what's the most important thing to curate. If we get a request sent from a user, we know that's an important entry to curate, and we will prioritize that as one of the first things we do. So if you see something that is under annotated, please let us know, and we'll look into it immediately.
Yeah, that's a good point. Yeah, the UniProt feedback from users is highly valued. Yeah.
Is there anything you want to add on this question, Stephanie?
Yeah, I think in the future we would like to annotate more AMR proteins that belong to the WHO priority pathogens. Yeah, that could be our future. But as Dave and Paul said, I think we valued the users' feedback a lot, and it probably will trump our priority.
Thank you. Just to add on this, so if someone wants to contact you to give feedback or a suggestion, they can do so through your contact page, right, on the UniProt website.
There's multiple links to the contact page. Every entry has a contact us link on there in several locations. Yeah, click on that. You'll be given a form. You can fill that in. It tells us what entry you're looking at when you click on that form, so we get all the information to know what you want us to update. And there's also general contact if there's anything general you want to let us know.
Perfect. Thank you. So I can move on to the next question that is about automated annotations. So someone is curious on how you can detect errors in automated annotation and how are these errors then corrected?
This is an ongoing process for us. We've got multiple automatic annotation pipelines, both curated by expert scientists and fully automatic machine learning ones. So these feed into each other, and you can often see when there's differences between the two, that flags something that we need to look into. All the curation that we do feeds into the automatic annotation pipelines. So when we curate a reviewed entry, that is what's used by the automatic annotation pipelines to be trained and annotate other entries. So when we do spot anything that seems obviously wrong, we try and fix it as quickly as possible. We do miss things. The automatic pipelines change, the data that they have changes. So again, if people see an error, let us know, we'll fix it. But it's something that we actively, when we curate an entry, we actively look at how that affects the automatic annotation pipeline.
Thank you. Any other speaker wants to add something on this, or just want to be sure everybody has the opportunity in case? Okay. So can move, actually, the next one is again on curation, and it's really related to the automatic annotation. So someone is asking, in today's LLM era, is UniProt using such approaches or planning to use them, and how, if there is a plan or there is already something?
I think it's another one I can take. So Paul mentioned in his presentation that for several years we've been working with Google DeepMind, and we provide LLM predicted or natural language model predicted names for proteins. We are continuing to work with them to expand that those predictions to other fields. And we're hoping in the next couple of months to release a next iteration of ProtLLM, which is the Google DeepMind natural language model, to add annotations to a lot of entries in UniProt. We're starting with a phased release of this because we want to see how people interact with it. And if that is received positively, we will pursue this further in the future and do a more comprehensive release across the UniProt database. So yes, we are actively looking at both using LLM to annotate, to help annotate unreviewed entries, but also using LLMs in our workflows to help us with annotation quality.
Yeah.
Thank you.
So kind of related to that is we're now beginning to use LLMs to assist us in our work. So behind the scenes, you may not be able to see it directly on the website in entries, but behind the scenes, we're using LLMs to help us. And it's been an interesting learning experience.
Okay, the next question is, okay, yeah, we're going out of the curation part. So one of the attendees is saying, thank you for the brilliant and well-explained presentation. I have a question about, after finding the proteins that are involved in antimicrobial resistance, so in UniProt, how could this then this lead to the drug discovery with the help of bioinformatic tools? So I think related to the prot, this would be what are the links maybe that you provide on the database that then could support users in downstream analysis or related to drug discovery.
I may give a start. It's a very difficult question. I don't think we provide any direct evidence to support drug discovery. But as Paul mentioned in one of the efflux pump, he summarized those chemicals that can inhibit efflux pump. This, if there is a drug that can inhibit the antibiotic efflux pump, that might be work to reverse the resistance. However, we don't really provide if the chemical is safe to use in human or not. We just try to gather as many literature data as possible to summarize useful information in the UniProt page. But if that's something useful for the community, we can keep this in mind and then put those information in, summarize those information from the literature in the UniProt entry in the future.
Thank you. Perhaps I have a follow-up question. I'm sorry, maybe Paul said it during the presentation and I missed it. I was wondering in the UniProt page of a given protein, if you're mentioning, for instance, this protein is binding or interacting with this chemical, then do you have links to other databases such as Campbell, for instance, where more information can be found for the specific chemical?
Yes, yeah, we do. There are links to both to KEGG and Campbell, and also things like DrugBase, which I believe is an NCBI resource. Yeah, so we do.
Thank you. So that perhaps can also be a first step for anybody
Yeah,
who's
that would
interested
be
in
another
Mhm.
route.
Thank you so much, Stephanie and Paul. Okay, so the next question is, can I use I guess, this in the future while doing research about cytogenotoxicity of medicinal plant used to treat cancer? It's a very specific question.
Yeah, I can't say I really know an answer to that. There are annotations for toxicity which we capture, but I have to say medicinal plants is not a topic I really know anything about.
Yeah, the expertise at EBI is more animal orientated and some bacterial. I'd have to have a look if we have a plant expert at SIB who might be more
Yes.
involved in this.
Yeah, I think more curation is done by our collaborators, our colleagues at the Swiss Institute of Bioinformatics, SIB, in Geneva.
Particularly for plants. Yeah.
Yeah, particularly. Yeah.
Thank you, Paul and David. Then we have a question, a more specific practical question, let's say. So one of the attendees is saying, I'm working in environmental metagenomics, and once I finish the pipeline, I have a data frame with lots of antimicrobial resistant genes with different coverage. Some of these coverage do not have active protein sequence. Do we have any way to have an automated check if the sequence contain active sequence implemented in Linux or in Nextflow? So I don't know if that's something that UniProt, I don't know if you have some information about this, or if this question does not fall in your expertise.
I'm not sure what the user means by active sequence. So does that ring any bells with you, Dave or Steph? No.
I'm
No,
assuming they
sorry.
might be saying active site or something to do with that.
Yeah.
An active protein. So there might be a
Yes.
lot of entries that
Yeah.
don't have an enzyme activity associated with it. So that
Yeah.
would mean that it hasn't been annotated with the enzyme activity.
Yeah.
But yeah, I think for the rest of the question, that's more related to programmatic access of the data and
Yeah.
a bioinformatics workflow, which isn't within the scope of any of us here. We do give some bioinformatics access webinars, probably a better place to bring this up.
How can we create a protein that we create through modeling? So I guess like predicting structure. How would you annotate or curate this? Compare maybe to somewhere where you have like an experimental structure. If I'm interpreting correctly the question, it rings a different bell to you.
Most of the data that we manually curate comes from the scientific literature. So if there's no, I mean, obviously the automatic annotations also add information about a protein to the entries for those proteins. But beyond that, I'm not sure how we could help with this question, if I understand it correctly.
Maybe I'm understanding it slightly different. They might be referring to how can we add sequences of synthetic proteins and get those created for function.
Yeah.
UniProt doesn't store sequences of synthetic proteins. We only store the sequence of naturally occurring proteins. So we do get people submitting synthetic protein sequences, but that's not within the scope of UniProt. So those wouldn't be curated within our database, if that's what the questioner is asking about.
Thank you, David and Paul. I don't know if you have, I mean, that's maybe not UniProt related, but since you're mentioning that many people are submitting actually to your database synthetic protein sequencing, would there be a better place to submit those out of your knowledge, or
Good
kind of
question.
redirect users somewhere else so you don't receive
I haven't found an alternative. There must be a database for synthetic protein sequences, but we haven't got one that we recommend for users yet.
Perfect. Thank you. Okay, so the next question is very much about UniProt and the role of the curator, basically your job. So someone is wondering, when someone is recruited into the brilliant team at UniProt, do you receive any form of training for the specific job? Maybe I can imagine many of you come from maybe a scientific background. I don't know if you were researchers before. So when you dive into this new job, is there a specific kind of training? And then they're also asking if there are any internship possibility at UniProt that you offer. If there is, I don't know, maybe something in the website that you can redirect them to.
I think it's a bit of both. So you do get training, but you bring a lot of your own expertise, especially when it comes to field-specific expertise. So you do develop your own way of working. But when it comes to the technicalities, how to do the curation, what the guidelines are, and the curation standards, yes, there's a lot of training when you first join, and it takes minimum 6 to 12 months to really get into the flow of curation.
Thank you.
And we've got a very diverse set of backgrounds. Some people have come directly from the lab. Some people who've done a PhD and then joined the team, having been in the lab for a while. Others who have an intermediate step, who've done some other kind of curation. So it's quite an interesting mixture, and really good to work with. Formal internships. Steph, what would you say about that?
Yeah, we would love to, but we are still thinking about. I just joined UniProt curation team. We are still thinking about the strategies. How do we crowdsourcing and make use of people's passion to help with our curation? But if you are interested in internship, I think we are happy to apply any internship together. And then if you really want to go into curation, I think our team is a good place to come to.
Thank you so much to the three of you for all the information provided for this question. Okay, then we have a comment. Thank you for the insightful presentation. And with another question specific to the UniProt KB entry. They say someone is asking, is it significant the fact that the UniProt KB entry can have 6, for instance, or 10 character accession?
Good question. I wondered it myself. It's probably got to do with the source of the submission when it was first accepted. So there's historical reasons. I don't know the exact difference between the 6 and 10. I don't think many of us do, but it's something I'm interested in looking into.
Yeah, there are also examples. I thought at first it was to do with how long ago those entries were created, but that's not the case, because in some cases when we've had to curate an entry from scratch, a brand new protein, say for example, that's been submitted by a user, sometimes those have 6 character identifiers or accession numbers. So I don't know.
Then we have one question about antimicrobial peptides. So we talked a lot about enzymes today. So someone is asking, the UniProt also annotated antimicrobial peptides and their bioactivity, etc.
Yes, yeah, we do.
Yeah. We get direct submissions of protein sequences for peptides as well. If they don't know the full length of a protein or whether the peptide itself is bioactive, we receive those quite regularly. So there should be some antimicrobial peptides present in UniProt already.
Thank you. I didn't see that. Sorry, because there is a question that just came in now. Someone is saying that may notice mistakes with protein names in the UniProt, and is asking if they come from genome annotation data. Because there are still mistakes with, it's more of a comment than a question, let's say. Someone is writing, there are still mistakes with protein names, but noticed that those have not been updated in UniProt, and was wondering if maybe they're automatically plugged in, I guess, from like genome annotation.
Yeah, we get a variety of incorrect or inappropriate names. Sometimes they're added by the automatic annotation pipeline, and we can fix those when we see them. So we need people to point those out. We don't always spot them ourselves, although we try and filter out as many as possible. Sometimes it's the submitter itself that has given that name. So a lot of the unreviewed Tremble entries will have a submitted name associated with it that was that's linked to that submission. We don't always have the time to look at all of these. So again, if there is something that is wrong, we need people to point it out to us. But if it's a submitted name, it's not something we can fix. That has to be fixed at the submission database. So if it comes in through a nucleotide database, then it has to be fixed at that source. And that usually has to be done by the submitter themselves. So it's a bit tricky to fix those.